নমস্কাৰ মই আকৌ আহিলো বায়োলজিৰ আৰু এটা নতুন ইউনিট লৈ আমি ক্লাস 12 ৰে ইউনিট এতিয়াও আজিও লৈছো দ্যাট ইজ সেক্সুৱেল ৰিপ্ৰডাকচন ইন ফ্লাৱাৰিং প্লান্টস আমি এতিয়া ভিডিঅ'ত এজনে প্ৰশ্ন সুধিছিলে 11 ৰ সিলেবাস নালাগে নেকি তেওঁৰ উত্তৰ হিচাপে মই কওঁ যে 11 ৰ সিলেবাস লাগিব কিন্তু যেহেতু তোমালোক ফ্ৰেশ 12 ৰ boards or exam লিখিছা গতিকে তোমালোকৰ কাৰণে হয়তো যিখিনি 12 ৰ মেটেৰিয়েল সেইখিনি খুব সোনকালে তোমালোকে আয়ত্ত কৰিব পাৰিবা বা তোমালোকৰ ৰিফ্ৰেশ হৈ যাব গতিকে মই সেইকাৰণে 12 ৰ খিনিৰে ষ্টাৰ্ট কৰিছো আমি 11 ৰ কন্টেন্ট খিনিও লাহে লাহে আৰম্ভ কৰিম গতিকে এতিয়া আমি আহো sexual reproduction in flowering plants so sexual reproduction in flowering plants if we have to talk about sexual reproduction in flowering plants then we'll have to know what is a flower. So flower is a reproductive part of the plant. Plant means which plant? That is the angiosperm. Angiosperm is also known as flowering plants. So another name for flowering plants is angiosperm. Angiosperm. Let us first look at the structure of a flower. So how is the structure of flower? The structure of flower is like this. It is made up of these parts. Okay, so this part at the base, this is called as pedicel, or this is also known as stalk. This is the thalamus. Thalamus is the swollen part, green part. Then the thalamus divides to become leaf-like structures. Those leaf-like structures together are called as calyx. And that calyx is holding the colorful part of the flower, which is called as the corolla. Inside the corolla, there is presence of some like two types of whorls are there. One is the male reproductive part, that is androecium. This is the male reproductive part, and the other part, this part is the female reproductive part, that is also known as gynoecium. So this is gynoecium. Gynoecium is the female reproductive part. Now each of these whorls, okay, these are called as the whorls. So each of these whorls are made up of some parts. What are those? The calyx is made up of certain parts which are called as sepals. S-E-P-A-L. Sepal. Corolla is made up of certain parts that is called as petal. Sepal and petal. Again, androecium is made up of some parts which are called as stamen. Androecium is made up of stamen, and gynoecium is made up of parts called as carpel or pistil. Carpel or pistil it is. So this is the structure of a flower. So now coming to each part, like the male reproductive part and female reproductive part separately. The male reproductive part, male reproductive part, which is also known as androecium. Androecium. So the male reproductive part, which is also known as the individual parts, are known as stamen. Individual. So this is one stamen. Now this stamen has two parts. This is anther and this is filament. Anther and filament. Now if we go in detail to look at the structure of anther of angiosperm or flowering plants is bilobed. Bilobed means what? It has two lobes. You can see two lobes in here clearly. So this structure is called as a bilobed structure. Now if we go at the cross section, like if we have to look at the cross section of how does an anther look like, then it looks somehow like this. It looks somehow like this. Okay. So what are these? These are microsporangium. These are microsporangium. Microsporangium. Another name for microsporangium is pollen sac. Pollen sac. So what are these? These are the pollen holding part of anther. It contains pollen. Each of these contain pollen. Thousands and thousands of pollen in each of the pollen sac. There is a single connective tissue in here which is known as connective. This is called as connective. This is connective. Now coming to the structure of this anther. This anther basically has 4 layers in total. 4 layers in total. 3 layers like this, and the last layer around each of the pollen sac. The outer 3 layers are epidermis, then the second layer is endothecium. Endothecium. This is endothecium. You can see this is the endothecium. Then the innermost layer is the middle layer. This is called as middle layer. This is middle layer. And the innermost layer, which is also called as tapetum. Tapetum. The innermost layer is the tapetum. Now you can see the tapetum is the innermost layer, and 3 layers are there which are present outside to this. Like 3 layers in combined, that is epidermis, endothecium and middle layer. These 3 layers are responsible for dehiscence. That means when the anther will rupture to release all the pollen grain, that process is called as dehiscence. Whereas the inner layer, which is the tapetum, will provide nourishment to the growing pollen grain. So this is the rough structure of anther. This is the rough structure of anther. Now anther will hold all the pollen grains possibly present in the plant. I guess all of you are clear with this part. Now we will go to how the pollen grains are produced. Actually, the predecessor or predecessors of the pollen grains are called as microspore. And how do the microspores are produced? Microspores are produced by the process called as Microsporogenesis. Microsporogenesis. How does microsporogenesis occur? So when I have previously drawn the diagram of anther, so inside the pollen grain, inside the pollen sac or microsporangium, there are sporogenous tissue. In this sporogenous tissue, there will be appearance of certain cells, and these cells are called as pollen mother cell. Pollen mother cell. This pollen mother cell will undergo meiosis division. It will undergo meiosis division. It will produce 4 cells in total. It will produce 4 cells in total. As we all know that meiosis process will always result in production of 4 daughter cells. So same way in here also there are production of 4 daughter cells. These daughter cells are called as microspores. Microspores. Now these microspores, they will eventually dehydrate, they will lose water, and then they will become powdery, and each of them will form a pollen grain. Each of the microspore is ultimately going to produce a pollen grain. So this is the pollen grain. Now pollen grain, what is the function of pollen grain? Pollen grain is the male reproductive part which is going to fertilize the female part, and then there is going to be the production of the zygote, and that zygote is eventually going to form seed, and the ovary is going to form the fruit. So if we go to structure of pollen grain, how does a pollen grain look like? A pollen grain is, when mature, a pollen grain look somehow like this. Somehow like this. And what are these parts? This part are so pollen grain basically has two layers. Outer layer is called as externa, and the inner layer is called as interna. Externa and interna. Externa is made up of one of the toughest substance available on surface of earth, that is sporopollenin. Sporopollenin. This is one of the very tough substance available. Sporopollenin. That is sporopollenin. And interna is made up of substances like cellulose and pectin. These are very common polysaccharides available on in nature. This is cellulose or pectin. These are going to form the interna. Now inside this it has cytoplasm, and we can see two cells. Very clearly we can see two cells. One cell is called as vegetative cell. This is vegetative cell, and this is the generative cell. This is generative cell. So this is vegetative cell and this is generative cell. Now this vegetative cell is going to provide nourishment to the generative cell, and generative cell is going to divide into two. It is going to divide into two before it fertilizes any of the female gamete. It is going to produce two. It is going to be there are going to be two generative cells. Now you may wonder, what are these? These are nothing but these are germ pores. These are called as germ pores. What are germ pores? Germ pores are basically gap on the sporopollenin, so that the pollen tube can easily come out of the pollen grain. Because sporopollenin is very tough substance and it cannot be broken down by any enzyme or any chemical. So naturally there are gaps in the outermost layer, that is the sporopollenin layer, so that the pollen tube can easily come out of the cytoplasm. Again, pollen grain has certain, pollen grains are not always good and pollen grains are not always bad. We can say some of the pollen grains can produce allergic reactions like hay fever, allergy, bronchitis, etc. And some of the pollen grains are also at times pollen grains are also used as nutritive supplement. A nutritive supplement, pollen grains are used for better performance of the athletes, and also for the race horses. Then pollen grains have certain period within which it can germinate, and that is called as pollen viability. Pollen viability can vary from species to species. In case of plants like wheat, rice, etc., the pollen viability is only 30 minutes, whereas in case of plants like rose plant or potato, in those the pollen viability can last for up to a month sometimes. And then the pollen can also be stored at very low temperature. The temperature is minus 196 degrees Celsius in liquid nitrogen. It can be stored in liquid nitrogen. Liquid nitrogen can be stored at minus 196 degree Celsius. So this is about the pollen grains. Okay. Now coming to the female reproductive part, nothing to be much to be changed in here. Just I'm going to change this to female reproductive. Female reproductive part, and then the female reproductive part is known as gynoecium. Gynoecium. Okay. Now each of the gynoecium, like I am going to draw. The unit was, as I have told you earlier, units are carpel or pistil. So I am going to draw a carpel or a pistil at one go. So this is a carpel or a pistil or pistil. This is carpel or pistil. Now looking at the parts of the carpel or pistil, this part is stigma. This is stigma. This part is style and this is ovary. Stigma is the landing platform for the pollen grains, whereas the style is the connecting part between the ovary and the stigma. What does ovary contain? Now let us look at how does an ovary look like. So ovary, so this is the ovary. So if we look at the ovary, the cross section of ovary, how does it look like? It is like this. Cross section of ovary is this, and it has certain chambers in it. These are the chambers, and each of these chambers contain certain structures in it. So this chamber is called as locule. This is locule, and the inner part, the inner structure is called as ovule. Locule and ovule. Locule, like the ovule, is attached to the lobule. Locule is attached to the ovule with the help of a structure called as placenta. Locule is attached to ovule with the help of a structure called as placenta. Now let us look at the ultrastructure or the enlarged view of the ovule. So the ovule looks somehow like this. Ovule is like this, and then in the ovule is connected to the locule with the help of funicle. The placenta is somewhere in here, but the funicle is the connecting link. Funicle is attaching the ovule to the locule. Then the point where funicle get attached to the ovule is called as hilum. Hilum, hilum. Then coming to this part, this is called as chalaza. Chalaza. These are the parts which are also covered, which are covering the the ovule. These are called as integuments. Integuments. These are integuments. This is the outer integument. This is the inner integument. Inside to the integument there is a kind of cell which is called as nucellus. Nucellus, and this part is called as embryo. Embryo sac. This is, and this part, this gap is called as micropyle. So I want all of you to have a good look at the structure of ovule. So this is the structure of ovule. You can see this is the embryo sac, this part is the chalaza, this opening is the micropyle, this part is called the spunicle, where it has attached to the ovule is called as hilum, then the inside tissue is called as nucellus, and this whole structure is attached to the locule. Now if we look at again, if we have a closer look at this part. So how does it look like? I'm going to draw it. So we can see here there are 7 cells. We can see these cells at the chalazal end are called as antipodal cells. These are the antipodal cell. This is polar nuclei or central cell. This is called as central cell. Then this part, this cell, the surrounding cells are called as synergid, and this is the egg cell. Just have a good look at the structure. These are the antipodal cells. This is the polar nuclei, and these 2 cells are synergid, and the middle cell is the middle cell is the egg cell. In the synergid, there are presence of certain finger-like projections. These projections are called as filiform apparatus. So what is the function of filiform apparatus? Filiform apparatus is basically responsible for guiding the pollen tube towards the egg cell. That is the function of filiform apparatus. Now going to the process of megasporogenesis. Now for megasporogenesis, again I'll have to draw the structure of ovule in rough. I'm drawing because previously I have already drawn it. So in here there is sporogenous tissue present. So from the sporogenous tissue, one cell is going to enlarge and it is going to produce megaspore mother cell. So here we are talking about megasporogenesis. Megasporogenesis is production of embryo sac where the female gamete is there. That means the production of female gamete. Also we can say production of female gamete is megasporogenesis. So there is inside this ovule there is production of megaspore mother cell. This megaspore mother cell is going to enlarge and it is going to look different from the surrounding cells. Then what is it going to do? It is going to divide. Again it is going to divide to form 4 cells. Out of these 4 cells, 3 cells will degenerate. 1 cell will remain and this cell will enlarge to form the embryo sac. And the 1 nuclei will divide to form 2, 2 will be divided to form 4, 4 will divide to form 8 nuclei, and each of the nuclei will go to 4 nuclei in here, 4 nuclei in here will go, and then from here one nuclei will migrate to the center, and then here 1 cell will form, here 1 cell, here 3 cells in total, and again in here there will be formation of 3 cells. This diagram I have already shown you before. So this is the embryo sac. So this is how the megasporogenesis take place. So next, what are we going to see? We are going to see a phenomenon called as pollination. Pollination. Now, what is pollination? Pollination is nothing but transfer of pollen grain from the male gamete to the female part of the same flower or different flower, okay, of the same plant or the different plant. That is what is called as pollination. Now, I am going to discuss about the types of pollination. What are different types of pollination? The pollination, the first type is autogamy or self-pollination. Autogamy or self-pollination. The second one is geitonogamy. Geitonogamy. Third one is xenogamy. Xenogamy is also known as all of us know, that is cross pollination. Xenogamy or cross pollination. Now, this xenogamy or cross pollination, xenogamy or this one. First let us discuss about this one. Autogamy or self-pollination. Autogamy or self-pollination. Again, autogamy or self-pollination means when the pollination is taking place inside the same flower. It is called as autogamy or self-pollination. But there are two different types of flower in one kind of plant. That is, one is chesmogamous flower, and the other one is clistogamous flower. Chesmogamous flower and clistogamous flower. What is a chesmogamous flower? Chesmogamous flower is a normal flower where the pollen and the anther and pollen, everything, anther, pollen, pistil, everything are open, and there are scope of also cross pollination. But the clistogamous flower is the closed one where the pollen and the pistil are enclosed, and thus only self-pollination will take place. This kind of these flowers are mostly seen in case of plants like flowers like oxalis, viola, pomelina, etc. Geitonogamy, it happens when in the same plant, if there are two flowers in a same plant, and from one flower anther is transferred to the pistil of another flower. So that is called as geitonogamy. Geitonogamy is actually it can be called as autogamy only, but practically if we look at, then it is xenogamy or cross pollination. Now coming to the last one, that is xenogamy or cross pollination. Xenogamy or cross pollination, we have like different pollinating agents. We have pollinating agents. We have. So pollinating agent, this one, the first pollinating agent is abiotic. Abiotic, we have air and water. Air is anemophily, and water is hydrophily. Then we have biotic agents. Now biotic agents, there are many biotic agents. In here I am writing biotic agents. Animals, it can be animals. If it is animal, it is called as zoophily. If it is insect, it is called as entomophily. Animals, it is zoophily. Insect, it is entomophily. Sometimes it can be snail. If it is snail, it is called as malacophily. Malacophily. Sometimes it can be also bird. If it is bird, it is called as ornithophily. Ornithophily. So these are animals, insects, snail, birds. There are many biotic agents which can cause pollination. So according to that, there are different types of names. One important thing I would like to tell you is that you see abiotic factors, for the abiotic factors, air will require the pollen grains to be light and non-sticky, and water needs the plant to be water plant, like water in the water growing plant. But sometimes we might be confused, like in case of lotus or lily, etc. We think that they have water pollination, but actually they don't have water pollination, because even if they grow in water, but their flower is outside, like it is much above the water level, so there is no pollination by water in them. Water pollination happens in case of certain grasses like sea grass, zostera, etc. In these kind of plants we can see the water pollination. In biotic agents, the flowers which are pollinated by biotic agents, they need to always have something, like they need to have a very foul smell, they need to have very good smell, sometimes they need to be colorful, because the animals, birds, insects, they need to be attracted to the flower for the pollination. So for the attraction, they need to have a particular property. That can be a foul smell, that can be a good very nice fragrance, or it can be sometimes different types of colors. Then coming to the next one, that is what happens after fertilization. Now what happens after fertilization, if we look at the this diagram, you see this is the diagram, this one. Say the pollen tube, this is the stigma, suppose this is the pollen tube will come and it will enter inside. It will release two cells, two generative cells, two germ cells, as I have already told you. Two germ cells it will release. One germ cell will fertilize the egg cell, and one germ cell will fertilize or fuse the central cell, or polar nuclei. This is going to produce zygote. This is going to produce zygote, and this is going to produce primary endosperm. Primary endosperm nucleus. It is going to produce primary endosperm nucleus. Now this primary endosperm nucleus in future is going to produce it is going to produce endosperm. Now what is an endosperm? Endosperm is a nutritive tissue for the growing embryo. It is a nutritive tissue for growing embryo, endosperm. So this is a triploid cell. Remember this. This is a triploid cell, and this is a diploid, twice N, because there are already two nuclei present. This is the third nucleus from the germ cell, so it has become triploid, twice N, and this is the diploid. Zygote is the diploid. Zygote ultimately is going to produce embryo, and from the embryo there is going to be production of seed. So you see there is production of seed, and seed can be of two types, like monocotyledonous seed and dicotyledonous seed. What is a monocotyledonous seed? Monocotyledonous seed is somehow like, if we take example of a gram. আমি যেটো বুট বুলি কওঁ বুট chana. So a gram, if you remove the outer cover, then the chana looks somehow like this. So this is one cotyledon, this is another cotyledon. So this is called as dicotyledonous seed. Dicotyledonous seed. But if we look at maize or if we look at rice, you cannot split it into two parts. This is called as monocotyledonous seed. Monocotyledonous seed. This is monocotyledonous seed. Now, the seed is covered by, seed will be formed by the egg and the fruit. The seed will be covered by fruit. The fruit will be produced by the ovary. When the fruit will not produce from ovary, but it will produce from somewhere else, it is called as false fruit. It is called as false fruit. What did I say? That when fruit is developed not from ovary but from other part, then it is called as a false fruit. Example of false fruit is apple. Example is apple. Now, sometimes what happens, in case of certain plants like banana, the flower, there is flower, but the ovary grows without fertilization to produce fruit. Such process is called as parthenocarpy. Parthenocarpy. Example, banana. So what did I say? When the flower do not have fertilization or the flower do not undergo fertilization, but the ovary directly form a fruit, then it is called as parthenocarpy. The process is called as parthenocarpy. Then we have last two concepts. One is apomixis, that is formation of seed without fertilization. Parthenocarpy is formation of fruit without fertilization. Apomixis is formation of seed without fertilization. And polyembryony means when in one when within a single seed there are multiple embryos present, then it is called as polyembryony. So that is apomixis and polyembryony. For this I will provide the notes very soon, and also I am going to send you a MCQ, a group of MCQ. You try to solve those MCQs, and if you have any doubt, then you can ask me, or regarding any doubt on YouTube comment also, you can ask me whatever you have not understood. So for today, this topic I'm over with. Thank you. See you soon.